Good morning, everyone. Thank you for attending this virtual ACCP's webinar. My name is Salil Painse. I'll be leading this roundtable discussion today on an overview of QSP and PBPK modeling and how we can incorporate more biology into our modeling exercises in the pharmaceutical space. I work as a quantitative systems pharmacologist at Nuventra, and I'll go through this presentation, give you a brief overview of what I think the approaches are that benefit from adding more biological constraints to our modeling systems. And then after that, me and my colleagues over here would be happy to take some of your questions. To just get started with this, I know this is sort of this is something that all of you know, but I just like to start off with some basic understanding of the terms. PK and PD are the two important facets of drug action that we study here. PK deals with drugs exposure by characterizing its absorption, distribution, bioavailability, mechanism, metabolism, excretion as the drug enters the body and leaves it over a period of time. Whereas PD of the pharmacodynamics component mainly deals with the response that the drug actually elicits in the body, either through biochemical or molecular interactions or any of these different mechanisms that happen. So you can see largely the PK part of it is described by different processes that happen in the green circle below, and then the blue circle highlights the biological effect of the drug you're taking in. So the PK of the drug is traditionally described as a time course over a period of plasma concentration over a period of time. We have different values that we take out of this concentration profile, namely Cmax, AUC, half time, elimination, half-lives. We have the minimum concentration that the drug reaches. All of these different parameters help us inform what the drug is doing in the body. And so the basic PK, the single and multi-compartment PK parameters, PK models really take all of these things into account to come up with a way to model the graph that you saw in the previous slide. And so over here we have a central compartment within which dosing occurs. The drug is introduced into the central compartment either through IV dosing or through oral dosing. If it is going to go through oral dosing, then it obviously has to go through an absorption phase where it needs to make it to the central compartment from the gut lumen, and there are some biological effects that need to be considered there. And then from within the central compartment, the drug can leave the central compartment through multiple different routes. The clearance can be renal, the clearance can be metabolic, pulmonary, fecal. It can also be excreted through other means. All of these different types of clearances are additive, and which is what essentially a PK model is trying to simulate. You add all of these clearances up, you calculate the drug coming in, and whatever is left in the body is what gets distributed through your volume of distribution. Now, sometimes a single central compartment is not enough to completely explain the kinetics that we see in a concentration time profile. And so in those cases, we can add multiple peripheral compartments to the model to explain the elimination happening at different rates than what we would expect from a pure central compartment. And this traditional approach works really well for a lot of cases. But then every now and then we run into cases where we really need an added description of the biology behind why we are doing, why we see a particular time concentration profile. Some of the examples of that would be we want to extrapolate from a non-clinical species into humans, and we have certain non-linearities in the dose response in humans or in the non-animal species that we are not able to simply extrapolate using a standard PK model. We may need to extrapolate between different ages, and traditionally the way that is done is using allometric scaling. Of course, allometric scaling assumes that your entire IDME process is linear with respect to body weight, which is not always true. And in those cases, we may need to better describe the differences in biology between a pediatric population and an adult population to better understand what the Cmax and the AUC should be. Similarly, the absorption phase may need to be better described to investigate portal of entry effects. A classic example of this is the effect of food on drug absorption. And this is an emerging area where PBPK models have been used in the past and are continuing to be used even more. Traditional PK models, the assumption that we have is that the system is well distributed, and the central compartment really is an estimate of uh, the concentration of the drug in a large volume of distribution that is spread throughout the body. Uh, However, sometimes that assumption isn't necessarily true, specifically in cases where diffusion isn't the only process that is driving the concentration of the drug entering the amount of drug entering specific target tissues. And target tissue concentration at the end of the day is our key to understanding what is happening, and that is the effective concentration of the drug that our target tissue is seeing. And in cases like these, we may need to go to PBPK modeling to better explain what physiological processes are leading to these differences in target tissue concentration versus systemic circulation concentration. Classic examples of these are concentration in diseased tissues like tumors, where you don't have all the normal physiology happening. Transport across blood-brain barrier, transport, or what happens when uh, drugs are exposed, when the drugs are given to the mother but the fetus is then exposed, and we need to understand whether the drugs are able to transport freely and what is the what is the population concentration of the drug in fetus. And then, of course, this is the final is a classic case where PBPK models are used are the potential for drug-drug interactions in enzyme and transporter activity. And we'll get into that in a little more detail a bit later. But these are not the only applications of PBPK models. Think of PBPK models as a way to describe PK in your systems, where traditional PK models are not capturing the complexity of the system well enough. And we know something about how the biology is actually reacting and creating the PK that you see in your non-clinical studies or that you see in some early clinical studies. That is where we can then incorporate biological detail into our PK models, and we convert them into a physiologically based pharmacokinetic or a PBPK model. So how do we do that? We do that by essentially expanding the distribution, the central compartment model into individual compartments that represent specific organs in the body or specific tissue components in the body. So this is an example of a PBPK model. Not all PBPK models are the same. In fact, it's highly recommended that you build a PBPK model that is specific to your application, that limits the overall uncertainty that you're building into the model. It also helps with making sure that you are keeping your models parsimonious and you understand what is going on. But in this case, for example, we have a target tissue that we are interested in investigating, and we are going to expose the drug is taken orally. That's why we have an oral exposure route. And the GI lumen then feeds into the liver through the hepatic portal system. We don't really expect there to be a lot of intrahepatic circulation with this particular drug, so that component is included in this model. So all of these different decisions that go into making the PBPK models are driven by your knowledge of what the drug is expected to do, what data you have to validate a given model, and what you're aiming to get out of it. But essentially, think of this as an expanded PK model that allows you to incorporate more biological relevance and allows you to do extrapolation in a much more biologically detailed manner. So to extrapolate, to use these PBPK models for extrapolation, the problem statement remains the same. We have an exposure to a dose in a source population like adults, or in a source species like, let's say, rodents or dogs, and we want to extrapolate it to our target population or target species. So the way to go about doing this when you're doing it with a PBPK model is you first parameterize a PBPK model for your source population or species. So if I'm starting with rat data, I parameterize a PBPK model. I input all of the metabolic clearance parameters that I know about my chemical, and I validate the model against known non-clinical experimental data. What that tells me is it gives me confidence that I'm defining and describing the physiological processes that the drug is doing in the non-clinical species effectively. Now, the next thing to do would then be to extrapolate the target population to our target population. Traditionally, this is done by pure allometric scaling in PK models. In PBPK models, we don't really have to do allometric scaling because we have individual tissues and organs that represent different compartments in which the drug can distribute. So the way we achieve this in a PBPK model is instead of using physiological parameters that represent a rat, if we are doing a rat to human extrapolation, we will input in physiological parameters, liver weights, blood flows, tissue fat concentrate, tissue fat proportions, partitioning between tissue and blood, all of these values for human population. And we will also incorporate, if we have access to in vitro data for metabolism, in vitro data for partitioning, we will also incorporate that into our model. So now we have our model parameterized for our human target species. Now, once we have this model parameterized, we can then run the model simulation to actually get our exposure to dose in a target population or a target species, allowing us to perform the extrapolation. And this loop can be repeated over and over again to add on more extrapolations as we need. And this has been done fairly regularly in the toxicology world. There are quite a lot of examples in the clinic, in the pharmacology realm as well, of the species or the age-based extrapolation approaches to PBPK models. These are some of the examples of the interspecies extrapolation, extrapolating from mice to humans, extrapolating from rats to humans. Uh, all of these different all of these different publications used PBPK models because there were certain non-linearities observed in the responses uh, in in first stage of clinical trials or in non-clinical data, and they thought that it would be better to use a PBPK model for doing these extrapolations. What they also were able to then, when you're doing PBPK modeling, you're able to pull in more data from in vitro sources. So for example, the first publication looked at measured in vitro clearance in microsomes and recombinant enzymes in both rats and humans, and then combine that information together to effectively extrapolate the data from non-clinical species to humans. The You can also use the same extrapolation approach to do adult to pediatric population. In this case, you take into account the enzyme ontogeny, that is how an enzyme matures over time, to scale the actual metabolism component or to scale transporter component or any of those things. So for example, in the graph that you see below, the y-axis represents age in years, the x-axis is a scaled morphine clearance in liters per hour per 70 kilogram body weight. And scenario 1 and scenario 4 depict two modeling scenarios in which either you take into account the ontogeny for the transporter and the enzyme implicated in uh, the pharmacokinetics of morphine, and in scenario 4 you take that into account, whereas in scenario 1 you don't take it into account. And as you can see, the black bars represent actual clinically measured data. When you take ontogenies into account, the clinical data is much better represented. And that is really the power of PBPK modeling, because if we were to do a simple allometric scaling between using just body weight, we would essentially get the same result as we get here in scenario 1, because that is morphine clearance that is scaled by body weight. So that is what we do when we are doing allometric scaling. So in scenarios like these, the PBPK models really shine through. Of course, we also have to understand the biological effects of drug exposure and not just PK. And traditionally, pharmacodynamics, uh, PK/PD models have been used to study these effects. The same PD components that we use in PK/PD models can also be used in a PBPK model to describe where the PK component comes from the PBPK model, whereas the PD component remains the same. And one classic example of this is DDI. We tend to think of DDI as a PK process, but in reality, DDI is really a PBPK PD model whenever you use PBPK modeling to do DDI, because the drug is actually causing an effect on the enzyme, causing either its induction or inhibition, which then leads to uh, that enzyme causing an effect on another drug that we are investigating. Similarly, there are cases where the drug uh, can reduce receptor expression, which leads to a lower effect down the line. In one of the studies that I have been involved in the past, we were looking at carbaryl and its effects on acetylcholinesterase depression in the brain. All of these are different ways to incorporate drug response, the biological response to drugs within PBPK models. And the way you do that is similar to how we would formulate a PKPD definition. We just add biological response components to our existing PBPK models and connect them through known induction or inhibition effects. Like with whether it's PBPK or PKPD, there are certain limitations with traditional PD modeling. All of our complex biomolecular interactions that lead to this biological effects are essentially distilled down to a singular rate of inhibition or induction, which means our underlying biological mechanisms are not well enough characterized to study them in detail. Now, for many times, this level of information is sufficient to what we need to achieve from our modeling studies. But going a step lower and actually incorporating that biological network interactions that actually are driving the PD that you see is important. And so the solution then is to start incorporating more biological relevance into these drug response pharmacodynamic models. And that is where QSP comes in. QSP, or quantitative systems pharmacology, essentially incorporates knowledge from multiple sources to build mechanistic models that are designed to do interactions between different entities in a molecular network. This knowledge can come from in vitro studies, in silico studies, transcriptomics. It can come from a, a large variety of experimental sources, and all of these together with a knowledge of those interactions can then be built in to create a network model where the inhibition or the induction of different proteins or receptors that we see or that we model in a PD system emerge out of these network of interactions. You're not specifically saying something gets inhibited. You have a network of interaction that leads to inhibition of an enzyme. And so the advantage of that really is now you have access to a much more detailed tree below just a PD component. That means you can actually test specific interventions at different places along this pharmacodynamic tree to find out how the response of a drug changes. If a drug is acting at node A versus node B, is it more effective, less effective? Does it have unwanted side effects? These are the questions that you can ask the model now. Similarly, you can flip that question around and say, what if in my virtual in my patient population, there is a chance that a particular interaction is not always as strong as it is in the general public, in which case you can also test those theories because now you have an existing framework to test these different clinical virtual and conduct different virtual clinical trials. Finally, you can connect these QSP models to existing PBPK or PK models in pretty much the same way you would connect a PD model to a PBPK model. So now you have the pharmacokinetic component, which tells you the distribution of the dose, and a much more detailed biological response components that allows you to test these different things. As you can use mechanism of action to specific for specific diseases, You can also use mechanism of action for specific toxicity pathways and see if your drug has toxicological concerns. I think a really good publication that I have found in my research is this by Bradshaw et al. It goes through a few examples of different uses of QSP, and it also talks about where they see opportunities, where they see criticisms of QSP, and where they see some more work is needed to fully understand these systems. So I'm going to quickly go through a couple of examples here. The first one is a paper by Klausnitzer and all, and what they did was they used a QSP model to identify novel interventions for treating Alzheimer's disease. And they combine their QSP model, which is the brain and the CSF model that you see up there, with their PK model, which was the plasma model that allowed them to then actually deliver their dose, their drug to the brain, and then perturb the system to come up with a place where they could, where a novel treatment option can be used to target sphygmolipid dysregulation. And the way they uh, ground truth at the model is there are other interventions out there that look into cholesterol biosynthesis and APP processing, and they used those models to actually validate their existing QSP model. In another example here, they used QSP models to conduct virtual clinical trials with specific patient populations, and they were able to identify why certain populations would be able to respond favorably to the drug that they were investigating and certain populations wouldn't. And this could happen, all of this can happen before you sort of enter into the clinical trials. So you can either structure the clinical trials so that you understand what you're going into and what you're going to expect out of it, or you can conduct a larger series of clinical trials with specific populations to further to get more out of your clinical studies. Those are just some of the examples of QSP modeling. QSP modeling and PBPK modeling in general can be used wherever we need more biological detail to answer our questions. And so to summarize, PBPK models sort of add more biological constraints to traditional PK models, allowing them to address certain challenges faced by traditional approaches. QSP models do the same thing for traditional PD models, and together we can use them uh, in certain scenarios and give us more detailed information about our drug. So with that, I would like to thank you for listening to my presentation, and we have a Q&A session right after this. So me and my colleagues here would be more than happy to answer any questions that you have on this or QSP or PBPK modeling approaches. Thank you.